Hello students, I am Atul Sharma. I am a faculty of anthropology, and today we are going to do the topic Hardy-Weinberg Law, very important topic in UPSC CSC optional paper 1 under the biological anthropology. Okay, biological anthropology. Okay, so in Hardy-Weinberg law, it is a very important part in the population genetics portion of the syllabus. Okay, so here what we are going to do today, first we will see the context. Also, then we will study the key concepts and terms. Then we will see the equilibrium or the law itself. We will see the assumptions behind the law, and we will see the applications. These are the 5 things that we will see in this lecture. Okay, so let us start with the context. So first, the concept itself, the Hardy-Weinberg law, comes under population genetics, and it talks about evolution of population. Now, when we talk about evolution, we should know what is evolution. It is descent with modification, as given by Darwin. We very well know he was the greatest evolutionist of his time in the 19th century. So he said that evolution is descent with modification. So every time there is a reproduction, when it goes from one generation to another, it leads to some modification, and we very well know that it depends on the characteristic traits of the parents. For example, parent 1 and parent 2, leading to an offspring 1. So you can see the parent 1 may be a male or parent 2 a female, and both provide their gametes and genes to create a offspring which is having traits of both of the parents. So you can call it this process here is fertilization, and this is the stage of generation 1. First generation, when parents meet, they reproduce a child which is generation 1, and when this child mates with another person, creating another offspring 2, creates generation 2. So this is how the descent works. Generation after generation after generation, there is a transfer of characteristic traits, and you can see the modification also. because there is a modification, because there is always a random combination of the characteristic of parents' traits into the offspring's traits. Then another thing that you need to know about this evolution of population is this evolution was provided by Darwin for individuals. He saw this in individuals as well as the population. Okay. At the same time, there was another thing that you need to know. We are talking about the basic concepts, the context we are dealing. Second is, in this is the Mendelian genetics. In case of Mendelian genetics, we very well know it was given by Gregor Johann Mendel. It was also given in 19th century, though discovered in 20th century. What he said, he gave 3 laws which we'll discuss little in brief. Law of dominance, law of segregation, and law of independent assortment. Okay, so here in the law of dominance, what he said, he did in his pea plant experiment. Pea plant experiment, and he did all the kinds of experiments with the pea plant because it was having large trait varieties, and it was very easy to self-pollinate the plant to get the true characteristics. Okay, he was used to experiment with these plants. Okay, we'll discuss Mendelian genetics in detail later, but here, for the sake of Hardy-Weinberg law, we'll discuss little bit only of this basic law. Okay, so law of dominance, that says 2 alleles of a trait then dominant will express. That's what he said in his law of dominance. That means if there is a trait of pea plant, maybe a dwarf plant, and a tall plant. So in this case, it was found that the tallness was a dominant trait. Okay, and how it was found to be dominant trait? Because it was expressing itself in the pea plant experiment. Okay, so it was a tall plant and it was a dwarf plant, which we symbolize with small t, and a capital T is the tall plant. Now in this we have the dominance of the tall plant, and it will be expressing itself in particular proportion in the coming generations. Okay, for example, if these two will be mating, and if there are 4 plants to be coming, 3 will be dominant forms and 1 will be the recessive one, which is not much of an expression. Okay, so that was the thing. Now, law of segregation. Segregation means the traits will be separate. The traits will be separate on each allele. Allele means nothing but a variant of a gene. So any gene may be a tallness gene, okay, or any kind of trait gene may have 2 or 3 or more variants, but in case of Mendel it was only 2 variants, because in his time he did not know all these variations, but he was sure that only one allele will be expressing on one trait. Okay, so tall trait will be only with one allele, so that is called segregation of alleles in the traits. And law of independent assortment, that means there is always independence, that which of the particular outcomes will have which of the traits. So it will be a kind of random assortment. Whatever you do, it takes its own decision which of them will be assorted. Okay, which one of them will get its own trait. Okay, so though there is a lot to discuss in this particular Mendelian genetics, but this is just the brief of these laws. So you now know 2 laws. One is Darwin's evolution and another is Mendelian genetics. Now let us move to some concepts that you need to know in case of Hardy-Weinberg law in population genetics. Because before discussing Hardy-Weinberg law, you must know few important concepts. So key concepts. Or terms. First is the allele frequency. Allele frequency and genotype frequency. Allele frequency and genotype frequency. So what is allele frequency and genotype frequency? You very well know allele is a variation of a gene. And genotype is the overall genetic character, okay, of an organism. Now these alleles could be in general dominant, recessive and or codominant. Okay, these are few characteristics you very well know, that dominant are those which win over the recessive in expression. Recessive are those which do not express themselves in general. And codominant is that where both dominant and recessive ones or the variant different alleles express themselves simultaneously. So that is the basic variation in a allele. Okay, so let us take an example for this. And another genotype is overall genetic character. Okay, so for example, if we are taking a trait M and N, M and N, okay, so in this it could be tall or dwarf, different kind of, any kind of you can take, okay, any kind of trait. So what will be the combination? These are the three major combinations. You can take these in 100 individuals, possibly a population of 100. Okay. Now in the MM, let us take there are 49 individuals. In MN there are 42 individuals, and NN there could be how many individuals out of 100? 9 individuals. Okay, so what will be the genotype frequency? Genotype frequency is nothing but the genotype divided by the total individuals. That is 49 by 100. 49 over 100, that is 49%. Similarly 42%. Okay, and NN as 9%, which is nothing but characterizing what are they characterizing? MM, which is the dominant trait. So it is homozygous, dominant homozygous. In this case there are two different ones, so it is a heterozygous. Heterozygous, okay, and you cannot say which one of them is dominant or not, so it is just heterozygous. And in this case it is a again homozygous because they are same ones, okay, but the recessive, because we are taking here M as dominant and N as recessive. Okay, this could be any kind of trait. Okay, for example, if you take human traits, example could be widow's peak. देखा है ना कभी? Have you seen some people have, especially at the center of their head, some extra hairs are coming up. Okay, creating a kind of V shape. So that is called widow's peak. So these kind of traits we analyze and find out in this case what is their frequency. Okay, so in this case you have these kinds of genotype frequencies, but in case of allele frequency we will take for individual ones. If you take allele frequency for M, allele frequency for M, it will be, how will you calculate this? Because there is M in this also, M in this also. So how do you do? There are two M's, so you will double the number. So allele frequency will be 49 into 2, 98. Okay, and single from this, so 42 in this case. So there it is 140. And for allele frequency, you very well know 100 individuals are having 2 alleles each. Two alleles each. Okay, that is the assumption that we have taken. So for this we have 200, because total number of alleles will be for the 100 individuals 200 alleles. Okay, so you divide it like that and you get 70%. You get 70% if you take a percentage out of it, right? You get 70%, and rest of it, the N will be 30%. Okay, for M it will be 70% and for N it will be 30%. Right? Now you understand very well about allele frequency and genotype frequency, because in case of Hardy-Weinberg law we will be dealing with these terms. Okay, let us move forward. Now let us move to the concept of population, because it is population genetics. So this term is very important to understand, that what is the population that we are going to study. Okay, because it is population genetics. So population will be the breeding population. Okay, breeding population, not just any common population. It is a breeding population. That means it will be having some characteristics, like it will be interbreeding within the population, interbreeding within the population. Then there will be, for example, it could be a village. Okay, village. Now then it could be a homogeneous population. Very important, homogeneous population, so that there is no differences in the population itself. Okay, homogeneous population. For example, religion, caste, language, all these differences should not be there, because that creates a kind of desired directed mating among the population. Okay, so but we will take homogeneous population in one village, having one religion, having one language, having one ethnicity. All these characters should be there. Then in general, %90 of population should follow this strict. Majority of the population should be following this strict. That means that there will be the breeding population breeding should be less than the population which is total population. Very important to understand that this criteria should be met. Okay, now let us move to another key thing that you need to know. Now some evolutionary processes that you need to know, that we will be dealing with in this concept. Evolutionary processes. Because why evolutionary processes? Because evolutionary processes are those which are going to change the population. They are going to change the population. So what are the major evolutionary processes? Let us talk about them. First is mutation. There are majorly 4 or 5 processes, but we will discuss only 4 processes which are more important from Hardy-Weinberg Law's perspective. Okay, so it is mutation. Mutation is what? You must have heard about this term in your class 12, or anyone who has studied biology. If one does not have studied biology, then it's okay, because mutation is not a very complicated concept. It is random sudden change in the genes. Okay, mutation, random sudden change in the gene, any kind, because we all have genes. Okay, so this mutation can occur in us also. Okay, any biological organism could have this mutation. Now mutation could be in what? About genes, and these genes could be of two types: somatic and germline. Okay, these genes which are in somatic cells and germline cells. Okay, so in the somatic cells, what are the somatic cells? These are the cells which are not responsible for next generation, that is your body cells. But germline cells are those which are gametes, or as we call sex cells. Very important. That, for example, your sperm and egg, all these are germline gametes or cells. Okay, so those cells are having chromosomes, and those chromosomes are having genes, and these genes face mutation, random sudden change in the genes. Okay, and this mutation can be of three types. It could be neutral mutation, it could be detrimental, and it could be beneficial. For example, neutral mutation could be any kind of somatic mutation, okay, which is happening in any part of the body, which is not going to be transferred into next generation. Okay, detrimental, neutral, for example, your ear, this is a neutral mutation. Widow's peak is a neutral mutation, it is not having impact on survival. On the case of detrimental, it could be any kind of lethal gene mutation. Okay, for example, thalassemia, this is a disease which is very detrimental. Okay, sickle cell anemia, it is a detrimental disease. Okay, Marfan syndrome, very detrimental disease. Okay, which is impacted by the gene mutation. Similarly, in case of beneficial, you have some particular, for example, in some cases, some of the mutations could be beneficial also. For example, there is color mutation in the moth. It is a very small insect which is in general of a white color, but in polluted environments it becomes white, it becomes black for its own survival. So that is a mutation which is beneficial mutation for the survival. Okay, so that is a mutation. Another process is genetic drift. Okay, genetic drift, as the word suggests, it is a drift, which is again a sudden and random change in the composition of population. Okay, and it could be due to various factors. Okay, for example, this drift could be due to natural disasters, etc. Any reason could be there, like COVID-19 or any kind of. Okay, this leads to change in the composition of population, leading to change in the evolution and change in the allele frequencies and the genotype frequencies. This is a possible thing there. For example, you can have maybe a loss of main population, maybe 3 insects of this shape and 3 insects of this shape. Okay, so in this case, if these two get killed for any reason, there is only one of this is remaining, and this is leading to overall change in the allele frequency and the mating patterns that will change, leading to resulting population and various other processes that will come across. Next is very important process, which is gene flow. It can also be called migration. Very important process, gene flow and migration.